So welcome to the afternoon session. Right before the posters, we have 3 exciting talks in this next session. So let me introduce the first speaker, which is Dr. Ma, who is from Eliquent Life Sciences, and she will give us a talk, Non-Clinical Regulatory Considerations for Developing Cancer Vaccine Therapeutics. Thank you.
Thank you. Thank you, Dr. Webster, and thank you, the organizer, for this opportunity to present here today. I really enjoyed the meeting so far because it's my first time. It's a small meeting, and it's really intimate. And I got a chance to talk to a lot of the audience today. It's really nice. And then I'm also really inspired by all those nice presentations and late-breaking discoveries or in clinical trials and then findings. So this is really nice. I really think it's like more people should join us next year. Today, the topic, as Dr. Webster has already mentioned, I'm going to cover more of a non-clinical considerations for cancer vaccine development. So actually some non-clinical studies. As a non-clinical consultant, so my primary duty actually is to assist all the investigators to make sure that their studies, they are in compliance with the regulatory authority regulations. So will ensure a very smooth clinical development to support all the clinical trials. One of my, one area, so it's like I'm going to use as an example is to use cancer vaccine as one example, just to walk you through what the non-clinical studies needs to be considered before like a clinical trial can happen. Here's the outline of my talk. And then part 1, I'm going to cover the overview of cancer vaccines. I think a lot of speakers has talked about it already, a little bit already, but I'll just give you a more systematic approach. So I'm going to provide a definition and different type of modalities of cancer vaccines, and also the general mechanisms of actions. And then in part 2, I'm going to use the mRNA vaccine as one example to cover that. So what needs to be prepared for the mRNA vaccine? So one of them is how to design the mRNA vaccine cancer therapy, and then the regulatory pathways that associated with this cancer vaccine, and also what the non-clinical program looks like, what the typical non-clinical program looks like. And also followed by, there are some special considerations that's, usually not associated with every vaccine, but in some cases. If time allows, I'm going to walk you through a case study and before I wrap up the summary. So what is a cancer vaccine? Cancer vaccine will be divided into 2 different types, as the prophylactic and the therapeutic. As Jamelia has mentioned earlier, prophylactic cancer vaccines, those are vaccines that prevent the tumor formation, development. And some of the examples, one of them is the human papilloma vaccine, and the other one is the hepatitis B vaccine. So those vaccines aims to prevent the infection that link to the eventual cancer formation. The therapeutic vaccine actually differs from prophylactic vaccines in the sense that therapeutic vaccine actually is to elicit immune system that against the existing cancer cells that's been already treated with the different therapies. So you can imagine it's like more challenging to eradicate those cancer cells. And some of the examples of US approved vaccines in this category that involves the ProVage that Dr. Brovsky talked about earlier. And also, this is the first one actually is a prostate cancer specific vaccine. It's individualized. And also the second one is the GM-CSF, which is more broad spectrum. So it's like can be used for treating various cancer. So before I get into the details of the vaccine, so just want you to be a little bit more familiar with the idea of how the development of this cancer vaccine came about. Actually, it was happened like more than 130 years ago, and then the cancer vaccine actually, the idea was really inspired by the observation that spontaneous cancer regression occurred after some types of infections. And then there was, since then, a lot of research going on, but not until very recently, in the past several decades, there's a dramatic kind of achievement milestone in this area. So the first is being in the around 1990s, there was a first identification of tumor-associated peptide that's being recognized by the immune cells that recognized as being the immunotherapy for cancer vaccine development. And then since then, there were a lot of peptide-based cancer vaccine research going on until 2010, when the US first approved the individualized dendritic cell pulsed prostate cancer peptides become immunotherapy for prostate cancer patient. And most recently, as you may aware, we have the mRNA vaccines becomes a very promising alternative for cancer treatment. And during around 2017 was the first mRNA vaccine in the clinical trial, and currently this is in the phase 3 clinical trial. So there are 5 different major categories of modalities of tumor vaccines. The first one is using the tumor cell as the entire array of tumor antigen. And then usually this is pulsed with, on the dendritic cells to elicit immune response. The peptide vaccine is more of a tumor-specific or tumor-associated peptides that administered in the presence of an adjuvant to enhance and elicit immune response. And the viral vaccine is make use of genetically engineered virus that express the tumor vaccine, tumor antigen, and then to immunize into the patient and then elicit a response that way. And then there are the DNA and the RNA vaccines that utilizing the genetic material encoding the tumor-specific antigens and making use of the host cell system to express the protein and then induce the immune response. There are a lot of advantages of mRNA vaccine So that I'm going to talk about later. But regardless of a modality of the cancer vaccine, the general mechanism of action they're very similar. So as probably you heard the great talks from this morning from all our keynote speakers. So regardless, depends on the type of vaccines you injected into the patient. So the route of administration will be changed, but ultimately, the goal is to activate antigen-presenting cells to present those anti-tumor-specific antigens, and they can bring them to the local draining lymph nodes and activate T cells. And while the activated T cells will be able to infiltrate into the tumor and causing cytotoxic effect, at the same time release more tumor antigens and then keep the cycle going until complete eradication of the cancer cells. This is really a simplified mechanism of action. The actual mechanisms involves a lot more different pathways. So as I mentioned earlier, so there are many advantages associated with mRNA vaccines. One being it's a simpler design and the shorter manufacturing cycle. And it's very antigen specific, tumor specific. So avoid a lot of non-specific reactions that could cause autoimmune disease. And one thing also is it does not integrate into the genome, so it's relatively safe. So as I mentioned earlier, there are several clinical trials going on with the mRNA cancer vaccines. And the most forefront runner is the melanoma vaccine developed by, co-developed by Moderna and also Merck, currently in the phase 1 right now. So, so how to design an mRNA vaccine? So usually when one design mRNA vaccine, it's usually to compare the either the genomic or proteomic sequences between the tumor cells and normal tissues, and to identify the tumor-specific antigens. And then using the computational analysis, that will really narrow it down to certain immunogenic epitopes that only specific to the tumor, and also be able to bind to the human MHC to be presented. As a result, those sequences will be encoded into the mRNA construct with addition of regulatory elements to improve translation and prevent degradation. So with the help of a nanoparticle usually for packaging, so the mRNA vaccine will be delivered into the cells, and hopefully it's in a very stable and very sound condition so that it can do their work later on. So in the US, actually, the mRNA vaccine is considered as a biologic drug product. So as a result, it's regulated under the Public Health Service Act, and is overseen by the Center for Biologics Evaluation and Research, which in short is a CBER. And then this is a part of FDA division. And then to get the vaccine to be approved, the applicant must to show that the vaccine is safe, efficacious, and also with good quality for the intended use. So before any clinical trial happen, the applicant needs to apply, submit the IND, which is called Investigational New Drug Application to the FDA. And then this document, actual package, includes 3 major areas: CMC, which is chemistry, manufacturing, and controls. And then non-clinical, which includes the pharmacology, pharmacokinetics, and also toxicology. And then clinical, basically, it's more of a clinical protocol and a lot of other areas, including questionnaires for the patient, IFCs, et cetera. So for non-clinical, basically, the applicant needs to demonstrate all those 3 areas in order to support the clinical trial and also the clinical trial design. So because mRNA vaccine is relatively a new modality, as a result, there is no dedicated regulatory guidance from health authorities so far to illustrate exactly what studies needs to be conducted. But we usually refer to the guidance, available guidance, ICH, WHO, and FDA guidance on relevant drug products. And sometimes we also refer to reference to the literature to devise the exact expectations from the agency. So for non-clinical considerations, what we have is for pharmacology studies. So in vitro, one has to show that the mRNA gene is expressed, the specific type of epitope is expressed in the transfected cell lines, and then this is a must show. You have to do the checkbox pretty much. And then in the in vivo proof of concept studies, one has to demonstrate this has to be done in the animal species that's relevant to human in terms of vaccine elicit immune response that's relevant to human. And during the proof of concept studies, the route of administration, the dose range, and regimen needs to be optimized. As a result, you can use that information to inform the design of a toxicology study and also the clinical trial study as well. So, for pharmacokinetics studies, actually most of the regulatory agencies do not have much of a pharmacokinetic requirement, but we do see a lot of companies actually they still add the distribution of the mRNA and or the protein and the duration of the protein. So that information will count towards the pharmacokinetic part of the study. And then if it's not provided, you could also justify why you think it's not necessary. And for this particular vaccine, it's okay. But definitely you need to provide justification or providing data either way. And then for toxicology study, again, you have to think about the toxicology study needs to be conducted in pharmacologically relevant species that species will elicit an immune response that's very similar to that in human. And also the optimized dosing regimen and also optimized route of administration needs to be applied in a toxicology study, which should be consistent what you're going to use in the clinical trial. Additionally, the formulation needs to be consistent as well, as much as possible to the clinical trial, so that you don't have to consider to have a bridging study later on. So basically, the mechanism of action should be also be similar in terms of how it's elicit immune response to the vaccine and how it's being protected in a human should be also reflected in the toxicology studies, in addition to the pharmacology studies. So as a whole, the safety evaluation will provide a really kind of a nice big picture to support the vaccine to be moved to the clinical trials. Oh, I advanced early. So besides that, this is there are other considerations as well. So if there's some additional, for example, if there's an excipient in the formulation that's considered as a new drug entity, so this particular excipient needs to be evaluated in the non-clinical studies to make sure that it's safe, and then before it can be applied into the clinical. So if there's a combination if there's a combination with the vaccine administration, for example, some companies would like to introduce cytokine or checkpoint inhibitor together with the cancer vaccine, then the consideration will be you have to provide rationale for this combination in the IND package. At the same time, if the combination drug product actually is at the early development stage, it's usually a combination of a vaccine and also the drug product will needs to be undergo the toxicology study again, unless you have a good justification that you think is not necessary. So for any drug product that will cause cytokine release syndrome, this is a big safety concern, and it needs to be evaluated. And then the final estimation of the starting dose will be really based on all the safety parameters and the data being evaluated, and also the cytokine release assay before the final proposal for the starting human dose can be accepted. So I think we still have some time, do we? Oh, we do. Okay. So I can walk through with you a case study. So this is one case where we have mRNA vaccine itself. It encodes the neoantigens of the tumor. And then at the same time, and then they want to introduce another mRNA vaccine that's encoding certain cytokine to really boost the immune response to the vaccine. So in this case, what we need to consider, so in the sense that if the new epitope if there's a new epitope formation, in this case, it's identified. And then there's one concern is autoimmunity. So usually we will, at the beginning of the mRNA design, we will just warn the the company this could lead to additional testing for potentially autoimmune in the future. But usually the sponsor, the companies are pretty good in terms of identifying the epitopes, like tumor-specific epitopes. But if that's happen, if there's ambiguity, we're going to tell the sponsor that something needs to be considered before you move into your development. And then another part is if the mRNA has the cytokine component in there, and then that could induce cytokine release assay syndrome. So that needs to be tested in the in vitro assays and also in vivo as well. So the final starting dose needs to consider both component of the vaccine and also the cytokine part of the mRNA to provide the starting dose for the clinical trial. So now we can just wrap up the conclusion. So what I've shown here today is cancer vaccine is designed to stimulate immune system against cancer cells. And then for non-clinical, the studies should demonstrate a robust efficacy in animal studies, and then using the same route of administration and also the dosing regimen as close to the clinical and toxicology study as possible to prevent additional studies in the future. And it's recommended, it's better to understand the distribution of the mRNA vaccine and also its duration, unless you have a good justification that you don't need to do those studies. And then for toxicology assessment, so basically this can be done just for vaccine, it can be done just in one species compared to other disease indication, probably you need to do into 2 different species. But for vaccine, if you have a good tox species, and then you're confident that it's representative of human immune response, you can just only do in one species. And the evaluation of a developmental and reproductive part is going to be part of a later on before, usually is before marketing application. So for initial studies, you do not need to consider the toxicology studies on the reproductive and the developmental aspect. So also, again, the specific non-clinical considerations for mRNA vaccine design needs to, it's really a target specific and population specific. So basically, it's really a case-by-case scenario. So with that, I think I'm conclude my talk right now. Thank you so much for your attention.
We do have time for a question. This one over here.
Okay, yes. Do you
In
need?
that slide, actually, you reference reproductive toxicity.
Mm-hmm.
And in that, I'm assuming you're referring to pregnancy as a condition?
Yeah,
Okay.
yeah.
We also know, though, and pharma doesn't usually take it into consideration, that PK/PD changes during a menstrual cycle. And I wondered how much that kind of detail is taken into consideration right now by the regulatory agencies.
Yeah, for right now, the regulatory agencies, this is not, we're talking not really about phase 1, phase 2, but it's like really before marketing. So right now they're concerned about the developmental and also more of different segments of reproductive. So it's like a early embryonic, and then whether it's EFT, so it's like a basically embryo-fetal development, whether it's affecting the fertility. So those are the more of a consideration. And as you mentioned, the menstrual cycle is part of the fertility. Those will be monitored as well, and then considered as part of the reprotox study. Yes.
So what do you see the authorities' requirement for future personalized vaccines? Because each vaccine will be unique
Yes.
in terms of toxicology requirements. So how do you see the regulatory evolving in managing the risk because it's cancer?
Mm-hmm.
What is your perspective?
Yeah, so that's a very good question, actually. So far, I think what we saw actually for vaccine, especially for oncology vaccine development, the regulatory bar is quite low. Because they're considering the patient population, their lifespan, and then the agency actually is willing to sacrifice some of the safety in order to for the cancer treatment first. So I see what you're asking about, because there's a differences between, so it's like an individualized, but I think so far the requirement is pretty much the same, because it's really important to get the survival rate up first before you consider other talks, it's like a side effects. Yeah. Mm-hmm. Yeah.
I have a question.
Yes.
So for vaccines, especially for prophylactic, demonstration of functionality of an mRNA candidate is important, especially in the early phase of development.
Mm-hmm.
What kind of animal studies do you recommend? Because sometimes it's difficult to develop the assays that show.
Yeah, I think, so, I mean, I think so far for the infectious disease, it's like a vaccine, the regulation from the agency is always basically the animal models has to replicate in terms of the immune response and the amplitude of the immune response in the patient. But sometimes it's really hard to do. I guess it's like for COVID, I think there are transgenic mouse model, but it's only replicate some of the human, it's like a effect. And then they usually use a combination of monkey and transgenic mice together in order to get a better picture. Yeah. Mm-hmm. Great, thank you.
Great. I think in the interest of time, let's thank Dr. Ma again.
Thank you so much.